Assalamu alaikum. Ladies and gentlemen, good morning. My name is Ahmed Al-Jabri, and it is my pleasure to welcome you all to the Food Safety Symposium 2023. Please join me in welcoming His Excellency Engineer Abdullah Al-Muaini, the Executive Director of Central Testing Laboratory in Abu Dhabi Quality and Conformity Council, who will now address a few welcoming words to the audience.
Getting old, I don't see that well. Ladies and gentlemen, distinguished guests, Assalamu alaikum and good morning. It is with great pleasure and anticipation that I welcome you to the Food Safety Symposium, an event dedicated to the critical intersection of food and environmental quality. Today, we gather as a diverse community of expert researchers, policymakers, and industry leaders to engage in meaningful conversation that transcend borders and disciplines. In today's rapidly evolving global landscape, the issue of food safety and sustainability, as well as environmental quality, have become increasingly linked. We are faced with the challenge of ensuring that the food we consume is not only nutritious, but also safe for our well-being. At the same time, we must be mindful of the impact our food production and consumption practices have on the environment. This symposium serves as a platform to explore innovative solutions, shares cutting-edge research, and foster collaboration that will shape the future of food and environmental quality. Ladies and gentlemen, in the food domain, we are not merely consumers, but stewards of our own well-being. The safety of what we eat directly influences our health and vitality. The symposium will delve into various factors impacting food quality, from production and processing to distribution and consumption. Our discussion will shed light on emerging risks, cutting-edge technologies, best practices, and regulatory frameworks that can ensure the integrity of food that sustain us. We will explore the complex links between human activities and the environment, seeking a harmonious balance that maintains biodiversity, protects environment, and promotes sustainable practices for the benefit of the current and future generations. Distinguished guests, as we embark on this symposium journey, let us approach our discussion with open mind, recognizing that the challenges we face require collective wisdom and collaborative efforts. Each presentation, every exchange of an idea, contributes to shared knowledge pool that has the potential to shape policies, transform industries, and positively impact lives. Together, we can drive positive change and create a future where food safety is assured and environmental quality is preserved. I extend my deepest gratitude to our distinguished speakers today, the event sponsors who have worked tirelessly to make this forum a reality, and of course, all of you, whether you are physically here or those who are joining us online. You are the vibrant minds shaping the dialogue today. Thank you very much.
Thank you, Your Excellency. And now please allow me to introduce you to Mr. Jesus Gomez, the General Manager of Waters Middle East, to address some opening remarks and introduction to the PFAS.
Well, in continuation to the warm welcome of His Excellency Abdullah Al-Muaini, I would like to address a few words on behalf of everyone. Ladies and gentlemen, marhaba and welcome to the Food Safety Symposium 2023. It is truly a pleasure to have each and every one of you here today. A special thanks to our co-organizer, the Abu Dhabi Quality and Conformity Council, and our co-partners, Gulf Scientific Corporation, the Global Food Regulatory Scientific Corporation, and the Arab section of AOAC International. Thank you. For the very first time, We are live broadcasting this event all over the Middle East and Africa, and we have about 290 participants, and that's why we are having a little bit of technical issues in the beginning. So I'm honored to stand before you as we gather together to embark on a journey of knowledge sharing, collaboration, and inspiration. This conference serves as a platform for professionals and experts from various fields to come together and exchange ideas, discuss emerging trends, and tackle the challenges that lie ahead. As we look around this room, we see a diverse group of people, each with their own unique experiences, perspectives, and expertise. It is this diversity that makes this conference so special. The power of bringing together minds from different backgrounds is boundless, as it enables us to unravel new discoveries and find innovative solutions. Over the next few hours, we have a remarkable lineup of speakers and exciting network opportunities. These will provide you with the tools, knowledge, and connections necessary to thrive in your respective fields and make a positive impact on the world around us. Whether you're an industry veteran, a young professional, a student eager to learn, or simply someone hungry for knowledge, this conference has something in store for everyone. On behalf of the organizing committee, I would like to extend my deepest gratitude to our speakers and volunteers who have worked tirelessly to bring this conference to life. Thank you. So let us embark on this exciting journey together. May this conference be a source of inspiration, a nexus of ideas, and a catalyst for change. I wish you all a productive and enjoyable experience. Thank you, and let the Food Safety Symposium 2023 begin. I will continue. Yeah. Okay, since I'm the first presenter on the first topic, the PFAS crisis, a new threat to human health. During this short presentation, I will try covering some basics of PFAS and share awareness for the better understanding of the subject. My colleague Rajesh will later dive deeper into it on how to test PFAS in various day-to-day products. What are PFAS chemicals? You may not realize it, but almost everyone has some levels of PFAS in their bloodstream because these products are everywhere in our everyday environment. PFAS is the umbrella term for thousands of different kinds of chemicals. They are known as forever chemicals. They are man-made chemicals used in thousands of different kinds of products. They are miraculous chemicals. You can't find a better coating, for instance, to make your jacket waterproof. They have made our life convenient in many ways, from the food you eat, to the sofa you sit on, to the rain jacket you put on when you go outside. Why are PFAS chemicals so dangerous? The problem is they don't degrade in the environment, and they are almost impossible to get rid of. They accumulate in soil, rivers, drinking water, and in the bloodstream of people who are exposed to them. And now PFAS chemicals are associated with a range of health problems, such as increased cholesterol level, low infant birth weights, effect on immune system, kidney problems, diabetes, cancer, and even diminished vaccine efficacy. How does PFAS contamination occur? While chemical manufacturers maintain that these chemicals are not harmful to the human health in the level detected in the environment. But of course, that really depends on where you're talking about. In some areas, particularly around the factories, the levels detected in the environment are incredibly high. We eliminate PFAS through loss of blood or in the urine. But one of the biggest ways to eliminating PFAS is through pregnancy and breastfeeding, which is incredibly worrisome. That means that children have some of the highest exposures because of mothers passing PFAS from their bloodstream onto their fetuses. And children are incredibly vulnerable to contamination because their organs are still not developing and their biochemistry is still evolving. Some scientists describe this as a ticking time bomb for future generations. This contamination might have never come to light if not for a plan to build a multi-million euro tunnel highway project that was due to pop out right next to a 3M chemical factory in Antwerp, Belgium. The plan required the digging up of tons of soil and river slit and being moved to different parts of Belgium. The decision to make all this soil to other parts of Flanders led activists to start scrutinizing what's in the soil. And two activists came across some documents that they requested from the state highway company and from Flemish regulators and discovered that the soil being dug up near the chemical factory was heavily contaminated with PFAS. The crucial question is, how can we avoid PFAS chemicals? Well, it is almost impossible to avoid them. PFAS are eliminated from the body primarily through urine, but this process can take years or even decades. Removing PFAS from water can be challenging, but several methods have shown promise, such as activated carbon filters, ion exchange resins, and reverse osmosis. Well, what definitely helps to avoid stain-resistant carpets and furniture and waterproof clothing and shoes. I would also say that buy drinks in a glass bottle and try to avoid plastic as much as possible. Do not use non-stick or Teflon-coated cookware. And what people always said, don't microwave plastic containers and try not to use microwave popcorn for kids. Some kinds of food packaging, especially from fast food restaurants, are linked to higher PFAS exposure. Use oral irrigators instead of dental floss, or use floss that is PFAS-free. These are just a few examples, and I think that our society nowadays has been moving away from plastic as much as possible. Let's try to leave the world better than we found it by reducing our environmental footprint and become more representative of the society we live in. I would like to end my presentation with a short video clip that shows how important it is to make a difference in life. And I hope the video is going to work with sound. Okay, let me try to find it. Okay, small problem. Okay. Just 1 second. So, okay.
What are you doing on the starfish?
Okay. Okay. We are having some sound problems, like in the...
You can't possibly make a difference.
Maybe is anyone on the mobile phone on the Zoom meeting? No. Okay. Well, I've removed the sound. I will explain the story behind. Volume out. Okay. The story goes like that you see this little girl throwing starfishes into the sea, and this jogger passes by and asks her, "What are you doing?" And she's saying that, "I'm throwing starfishes into the sea." And he shows hundreds of starfishes around the beach and asks, "Well, you won't make possibly a difference." And she picks up the next starfish, throws it into the sea, and says, "I make a difference for this one." And with that, to say that little by little, we make a difference. Not all the people, but somewhere we have to start. So thank you very much. We have some time to actually prepare the next presentation. So that's why it takes a few minutes to upload the next one. Next one is Dr. Watt, correct. Okay, I'll open the presentation to have it working. I'll let you come. Presentation, this is a PT. Okay, it now works.
Thank you, Mr. Jesus. Please join me in welcoming Dr. Wael Lameen, the Medical Director in G42, who will speak about the importance of wastewater monitoring program of Abu Dhabi.
Good morning, everyone, and thank you very much for the kind introduction. And thank you very much for the talk about PFAS, Jesus. It's becoming a very interesting topic in itself, PFAS. And so the title of the symposium was actually food. So how am I going to tie food with wastewater? So I think that in itself was a challenge in my head at the beginning. But the thought of the story is, if you remember in primary school, we all get taught about the water cycle. And the water cycle involves all the water that we produce and how it goes back to the environment. So there's a cyclical utilization of water. And with civilizations, humans congregated in smaller spaces, they realized the risk that it involves from getting humans living together. Apart from the domestic problems that wives and husbands would have, there's also other bigger problems from congregating humans in one place, in that they produce waste. And the waste that we produce is very likely to have downstream effects, whether into the water streams that we drink, or due to the infections that we have, or due to the discomfort that might come. And hence we invented, predominantly during the Roman Empire, sewage and sewers. And an interesting way of dividing the world into advanced and non-advanced, or high income and low and middle income, is dependent on their utilization of sewage and wastewater. So I think it's been on the forefront of many people's minds as to this relatively new science of wastewater or sewage epidemiology. And the famous story of wastewater surveillance started at around the 1850s with a certain John Snow, who was an epidemiologist based in London at the time, who realized that certain people were getting cholera. And then he tracked the causes of cholera and ended up realizing that it's from a water source, from a pump called the Broadway pump in central London. And by disrupting or fixing that water pump, the cases of cholera in London completely went down. And then in the 1940s, we started the polio surveillance program. So polio is an enterovirus, which is basically a virus that is shed in stool that causes poliomyelitis, which is a very debilitating illness. I don't think many people here have seen it, but where I come from in Sudan and Africa, we still had an ongoing polio problem into the 1980s and 1990s until this polio vaccination program started. Now we have a lot less. There's only a few cases reported in Pakistan last year. The last African country to have a case of polio is Nigeria, but the series is and the importance is that polio has been monitored over the last, now what, 80 years in sewage. In the 1990s, we moved on, 1980s, polymerase chain reaction was discovered, the utilization of polymerase, an enzyme which is heat stable, to amplify DNA. We have developed many methodologies since to identify pathogens based on PCR and based on the DNA content. The utilization in sewage was done in the 1990s. And the highest monitoring program in around 2010, also the Europeans and the North Americans started monitoring sewage or wastewater for chemicals, in particular for PPCPs and drugs in terms of chemicals. Those are the most monitored. And for infectious agents, primarily with the COVID pandemic, there was increased utilization of sewage monitoring for SARS-CoV-2. But the question is, why would countries and taxpayers invest so much money in monitoring sewage? So that there are many, many reasons. One of which is a lot of the sludge that is produced, or a lot of the waste that is produced, goes back into the agriculture cycle, which brings us back to the food cycle that we're talking about. So when you buy organic food, which is a very important thing that people tend to forget, organic does not necessarily mean free of toxins and free of chemicals. I think one of the points that Jesus mentioned is that, recommend consume organic. So the consumption of organic food is a complex matter, because organic, that means you will not be using petrochemical-based fertilizers like urea, but you'd be using probably organic fertilizers. The question is, what standard are we using to monitor these for toxins, PFAs, plastics, drugs, chemicals? The gold standard that we use is based on the EPA standards for monitoring sewage, and even those, not even within the United States, are agreed upon, in that each state has its own regulation for defining gray water. So, and the levels of metals and heavy metals and toxins differ from state to state, what is allowed and what is acceptable. This was published just a few months ago in The Lancet, I think. Sorry, I didn't put the reference here. I should have. So we started what we call, now there's a new science in itself that is growing very quickly called wastewater epidemiology. So that the science of understanding the risk associated with wastewater before and after discharge. And we're talking about raw sewage. The problem is there are more regulations about the effluent wastewater post cleaning in wastewater treatment plants. But from the time it leaves our bowl till the time you flush it, until the time it makes it to the sewer treatment plant, there aren't many regulations. And unfortunately, there's a lot of mixture with groundwaters. So that you also have to bear in mind, in certain places, we have septic tanks associated with well, and these are completely not regulated, mixing all of the groundwaters for us. So there's a relatively new concept, relatively new. It started in around 2010. It's called the One Health approach, whereby we start looking at the human health in a whole concept. So it started by looking at human health and animal health. So the links between zoonotic infections and human infections. So if you think, for example, all of you working in food labs, you test for certain bacteria, Salmonella, Campylobacter, Shigellas, E. coli 157, and toxin-related E. coli's. And this has been ongoing for 30 to 40 years now with the Food Regulation Acts. Each country has a Food Regulation Act that prohibits or limits the number of detected pathogens for foods, especially in outbreaks, and this is titled public health. But we also have zoonotic illnesses that are associated with animals, which are included with and have significant financial implications. If you think about the H5N1 outbreak over the last 3 years, bird flu, over the last 5 years, the cost of it only in 3 months in Spain alone, towards the end of 2022, was $3 billion in 3 months in lost poultry. So there's significant financial implications and economic outcomes. But recently, in 2016, there's a new concept called environmental health. So the cost and the importance of the environment as a whole. And by environmental health, we're talking about our green spaces, how much greens we have, but we're also talking about our impact as humans to the environment. So it's a two-way relationship, is how do we impact the environment and how does the environment impact us? And one of the most important things is our waste. How do we manage the waste that we produce, whether it's plastic waste, whether it's combustion, whether it's the sewage. And the sewage is important in that, if, for example, the PFAs that were mentioned by Jesus at the last talk included the what you have in your oxynorm, the cleaners, without mentioning a brand, but what makes your clothes whiter and cleaner. So these are all chemicals, but where does it end if you think about it? Where does this end? It ends in your sewage from the washing machine, and then you end up with a PFA. So what we are thinking about now is actually having interaction closer to the source. And apart from that, even the antifungals that are used to preserve the plants that we buy every day on the vegetables, so all of these fruits and vegetables that we import from Chile, cherries from Chile, kiwis from Australia, they're all covered with two things, wax and antifungals. And most of us, some of us don't wash them, we just eat them, but that would also end up in the waste. And even if you wash them to remove these, it's going to end up in your sewage. And that also has a downstream effect, whether it's into the fish or anything else. So if you think of the core of what we want to discuss here, the core is we want to have a better health for all of us. So that's the core of everything. And that is affected by the built environment and the social environment that we live out, which is affected by chemical, non-chemical stressors and systematic biological factors and activity and behavior lifestyle. So for one of us to be healthy, not if if the government tells you we will provide you the best health regime, build better hospitals, bigger hospitals, it does not necessarily mean that we will have better health, because there are other things apart from having hospitals that may affect health. Some of them are independent factors, like me going out and deciding to exercise. Some of them are without my ability to control, like the effectors, the chemicals that we require and are brought to us without our consent, or the systematic biological factors, which are gene and genome. So we need to, and a lot of them are affected by where you study, how you study, the dust outside. If you're watching the news, there's a lot of conversations about all of this and all the rest of it. And when we talk again, how does this start to wastewater? All of this ends up in wastewater. All of the chemicals we consume, whether we eat it, drink it, is usually either excreted in the form of urine or defecated in the term of stool, ends up in your cycle, whether it goes to the fish, to the agriculture, or back. And that includes pesticides, that includes your heavy metals, that includes your paints, that includes everything. It has to go somewhere. It doesn't just go to space. So it'll end up in the cycle, and unfortunately, it will end up affecting us. And hence, a lot of the projects that we're working on at the moment is trying to understand the use trends, trying to understand if there's a point of limitation or increased utilization at one point to have a highest effector method. So basically, sometimes you want the highest impact by identifying where the most utilization or most the waste is. And a lot of over the last 3 years now, nearly, we started a project with Abu Dhabi Quality and Conformity Council as our main stakeholders, along with the Department of Energy, which was decreed by the Executive Council, whereby we established probably the world's only dedicated sewage monitoring lab. We have developed a lot of new methodologies, some of which are unique to us here in the UAE with a lot of IP, by asking a lot of questions as to how to do it and how to develop these methods. So we asked very difficult questions because a lot of the methodologies were not updated for many years. If you look at whether standard methods or EPA methods. So we had to ask questions, for example, the volume of water for testing. We are using quite high resolution mass specs, some of it with our collaborators from Waters. And as we used a lot of robotics, we reduced the volume of samples to test, and we're testing for a big array of chemicals and microbes daily, more than 500 every single day targets on each sample from all around the city, from hundreds of samples daily. And not only, we're not the only ones doing this. These are just some posters that I got from a meeting I was in just a couple of weeks ago, the CTAC meeting. People are using it, looking at just the, looking at, this is not my work. This is from the Commercial Agriculture Experiment Station in the US. Again, the use of effluent and its effect on wastewater reuse for irrigation, which is what we're talking about. So if you look here in Abu Dhabi, they divide water into green, gray, and red. So the gray water is what they feed the plants with. So this in itself is cyclical and coming back indirectly or directly to us. Although it's semi-processed, it doesn't mean it's we have enough information about the effects later on downstream. There's a lot of work trying to predict the relationships and contamination, as I mentioned, with surface waters and deep waters. So again, this is a lot of work done by the United States Agriculture Agency. This is the EPA. They're looking at PFAs, as mentioned by Jesus over there, in wastewater. Yeah, so the variability of PFAs closer to sewage. And this is our own work presented at the same conference, but we were looking predominantly at the microbial landscapes and the availability of phages and viruses that infect bacteria and profiling microbial communities. And none of the work would be done without a very strong team, very collaborative and very supportive. So I'd like to thank each and every single one of them, and the trust that has been given to us by the leadership of this country to develop and work in this lab. So thank you very much. Thank you.
Thank you, Dr. Awad. And now allow me to introduce you to Mr. Rajesh, Senior Application Specialist at Waters, who will speak about the mass spectrometry for emerging contaminants.
You can come. Scope this down as well. Wait. Technology. Shit. Yes. Bertrand. Yeah.
Yeah. Oh, thank you. Okay. Thank you, Jesus. Okay. Good morning, all of you. Welcome to the symposium. Today, I'm just trying to give you some brief on what Jesus was talking about VFS. What here? Okay. I'll be just going through the actual analysis, actual scenario. Okay. That's very, very important to understand how we can go forward and analyze those compounds. Because, as you understand from Jesus' presentation, very clear that they are, everywhere, even in our blood right away. If you take our blood sample, we can analyze those compounds very easily. At the same time, our soil, water, whatever the food material, whatever the things, what you get, whatever the things from the nature, everything is now contaminated. Actually, if you look at this 1930s, these compounds they created just to help, to cover film. Like these compounds are actually for polymerization. They are actually they're helpful for so many other aspects, but they come into the nature and they entered everywhere. In fact, if you go to the polar bear, still we can see them. These compounds are everywhere present, where they're very high toxic, and they actually creates, they increase, cholesterol level. That's major issues, high blood pressures. Then there are, continuously if you are having, but they're actually persistent organic pollutants, cannot be destroyed. When you talk about them, so they are there everywhere, and they are bioaccumulatives, so that you cannot remove them. So how best we can see the concentration levels, and how is our water, everyday water we are drinking, right? How is our tap water? So there the aspects comes very important that everyone should have the understanding about these PFAS, which are actually hazardous. So we have just, if you see the guidelines, there are few guidelines already created by different regulations. Like EU, they have finalized that it's almost, we can say 0.5 to 0.1 to 0.5 microgram per liter. That is the levels what they have dictated. Now we need to see that these compounds are not so simple to analyze also, because if you have to analyze those compounds, it's very challenging. In fact, they are not so simple to identify. Why this interest has come? Because you see this, how much is hazardous? 50 billion euros. In fact, only for the Europe, that is the amount of spending happens on the health. So we definitely need to see that how we can regulate them and regulate our health. Where we can start? We can start with the EPA. Very clearly, they mentioned what molecules you need to be identified first, and what concentration levels you need to be estimated. And this, as I said, these compounds are not so simple. If you look at the structures, we can see them. They are not so easily ionizable or easily understood. And to what levels we can achieve, those things is very important for us to understand the levels what we are consuming regularly. And the limit is, you can see this 0.1, like, is a very low levels, in fact. And what is the right procedure? The right procedure is quadrupole. So quadrupole is one of the best technique what we can use it to quantify these analytes in different matrices. In fact, I will be also showing you some case studies which we have already analyzed in our demo lab. At the same time, we are going to, already few of our customers are very interested in doing this analysis in their labs. And I just show you what complete range of quadrupoles from water mass specs. And we have 2 mass specs are selected, selectively can be used for this analysis because you require high sensitivity. So one is this TQ-XS and another one is the TQ absolute. So TQ absolute is the latest model, but TQ-XS is a regular model which is already present in everywhere in Middle East. We also not only just end up with only the mass spec. What are the other tools required for that? So we have like, there's a separate kit was generated from Waters, and that actually can help you in starting up your analysis in your lab. So the complete mobile phase, whatever the contents are completely removed, and you can see that we are going to give the all accessories required. And there's a mobile phase separate bottles and even the liners and the column and then standards, everything includes in that. So what happens when you start this analysis, this kit is more than sufficient to initiate your analysis in the lab. And what it exactly does, in fact, because usually you can find even the content of PFAs, even normal, tubings, when you correct the tubings and the bottles. And you will see this, there is a peak at the blank without even having the standard. So when we just use this kit with this kind of, the tools, what I'm giving you, it actually helps you. There's no blank peak, in fact. So that's where very important that you need to have a 0 standard. It's very difficult to get, but of course, as much as low as possible, you need to start up with the analysis. And then we can use some of the other tools where we can make a standard addition method and then make sure that you do the quantification. If you go to the top end systems, what you are observing here, the direct injection is possible. Very simple direct injection is more than sufficient. That means you take water or soil, just make it a solution like in soil, and then take the filtration, filter it, and load it to the mass spec. That means you don't need to do any kind of sample extraction. Still, you will be able to analyze those compounds. Even in case of urine and the blood samples also, it's very simple that we can precipitate the plasma in case of blood sample. And we can also do the simple dilution of the urine and then directly inject it to the system. That means we can check any kind of sample analysis, is not required for this these systems to just do the analysis. So the processing time is very less, and run time is maximum. You can assign the run time maybe 10 to 15 minutes at the maximum, and quantify these compounds. So this is actually the protocol what we have created. One is a dilute and shoot. The other one is a solid phase extraction. And we have very low limits. We have, LLOQs. We can see that they're all, picogram per ml and nanogram per liter. That's what the concentrations we estimated in the different soil and as well as water. And the recoveries, we always see that as per the regulatory, 70 to 130% within that the recovery should be. And we have very good reproducibility data also. So when you make different matrices, we inject 20 times and see that the RSD within the 10% RSD, this molecule should come. And we have found some, take examples. We have taken the effluent, the groundwater, then surface water, everything we checked, in fact, and we found that some of the, the environment, we found already these contents are already present. That means definitely it is there. And when it is there in the polar bear, it definitely is there in our body also. And then we as per the regulatory, we have, the methodology can be adapted for every lab with a simple modification, the collection of the sample and the processing, SPE, whichever you want to make a modifications, you can definitely do that in that method. Okay, thank you. So in fact, we can use the SEP in Bioasys. We have the 2 different cartridges to concentrate the sample. In fact, if I want to have a lesser sensitive machine in our lab, definitely we can have lesser sensitivity, but we can definitely use these kind of cartridges to concentrate. And as per the regulations, it is already present, the methods. And if you see that there is like a WX, the cartridges which are already mentioned here very clearly as per the ISO guidelines. And we can definitely do this analysis, very simple processing technique. And the process is very simple. We have already created this one. I'm not mentioning everything here clearly, but it is very simple. Just pass through the cartridge, concentrate the sample, evaporate it, and you can make the concentrated and then inject into the mass spec. And we can see the limits we have already achieved in our lab, because these are the detection limits. And these are the recoveries. So SPE protocol, definitely we can follow. And of course, instead of that, if you have a good sensitive machine with you, yeah, definitely it is very easy for us to dilute and shoot into the mass spec. And this is the blank versus the sample with the different, samples like, surface water. And we also have a protocol like, how to enhance the system sensitivity by changing the source, like a unispray technology, where we increase the sensitivity or double or tripled in the same molecules. And this technique is patented and unique from Waters, in fact. Because if you see the unispray technology, we may not heard 5-6 years back, we don't know about this one. This is the new technology. It just exists and just started. In fact, this kind of ionization technique actually helps you the polar, mid-polar, non-polar, all different compounds. The scientists who are working in the lab, they must be knowing very well that they had to play differently with a different polarity of your molecules. And here, this source helps you all kinds of polarity. In olden days, if I say 15 years back, we had a technology called APPI, which is actually used for the non-polar or mid-polar to the non-polar compounds. And almost now it is completely, because APPI is not a sensitive technology, photoionization technique, but we come with the unispray technology, which actually essentially help you to increase your sensitivity at different polarity levels. So this actually why I'm insisting here that this technique was very good and useful for this PFA analysis. And of course, the latest model, which is a TQ absolute, actually makes you life easy in the lab, so that all scientists working in the lab, they don't want to do much sample extraction and spend their time in the extraction process. So definitely this, if you take a TQ absolute, it's going to change your, complete the processing time. And you just dilute as much as possible so that you have a less matrix effect and get the best data out of this. And it has come with a completely different technology, what we have right now. With a TQ absolute, we don't make a simple change. We make a drastic change. In fact, whatever the R&D work, what we do is like a positive and negative. It should be switching better, and it should be able to get the reproducible data. That's very important. At the same time, the negative mode, all these compounds, the PFAs goes in negative mode, and we require to have a proper sensitivity with that. Even if you want to have a positive and negative switching, that should happen systematically and get a great reproducibility. That's very important. So all scientists in the lab, they really essentially look forward for the, not only the sensitivity, the reproducibility. When you work with the polarity switching, definitely should perform in a different way. Just a case study, I just try to show you. This is how the real data in our lab, what we generated at the levels of 2 picogram per ml, almost say 0.002 nanograms. That's what we made the sensitivity, what levels it can achieve first. Then after that, the sample processing come into the picture. So whether you require a sample processing or not, regulatory says 0.1 picogram per liter, 0.1 nanogram per liter. It's almost 100 picograms. So we achieved up to 2, means almost almost 50 times lower sensitivity than the required. So from there, if you start the analysis, it's so easy for you. Even so, as per the regulatory, what it says, 5 times lesser than or 3 times lesser than the actual regulatory concentration. So if it is a 10 nanogram is your regulatory concentration, we just say up to, we have to go up to 3 nanograms or 1 nanogram at the maximum lower levels. 10 times it is actually. Or 2 times at least you should have your LOQ levels. So that's where, the sensitivity plays an important role, where you don't have much work on the, sample extraction process. And we have a very good linearity data from the different PFAs. These are all the things, I just want to highlight them, that this is the real data from the lab, what we generate. And the reproducibility, it has a great reproducibility data, what we have right now. You can see them very clearly. And this is not, as per the application note, it is just we created by ourselves. And then you can see that these are the transitions, and the system comes with that complete details. In fact, you no need to work or effort on doing the tuning part. If you say olden days, we used to do that, like, we used to take more than half an hour, 1 hour to tune the compounds and get the MRM transitions. But here we have a complete methodology which is posted and the system, so that you can just transfer that and start developing your analysis. So we have a complete chromatographic mass spectrometry as well as a chromatographic technique. And there's a gradient. All these details are clear. We also did extraction on the shrimp, the fish products. We have taken them and we did the analysis actually in our lab. And we see that as a great results by processing through WAX cartridges, in fact. So we have the complete procedure adapted our lab. And I'm going to demonstrate, in fact, next 2 weeks. I'll be in Riyadh to support one of the customer, and they started with the PFAs first. At the same time, I'm also doing this analysis in my lab, which is there in the Dubai demo lab. So you're all invited. In fact, if you want to come to lab and see how the analysis goes, and we have just procured the kit, everything is available, and we can analyze, and you can come with a sample if you want, definitely, and you can see that how the analysis will go. Will happen in our actual lab, actual scenario, in fact. And we have complete reproducibility and linearity, reproducibility data for most of the compounds which are generated, right? Like Waters is going to provide you not only the requirement for the analysis, if you see the standards, the first initiated, the 20-25 standards we will be giving you, so that you can start up this, and then we can procure few more standards from outside, and then we can add up the same analysis and do the analysis very easily on our mass spec. So this is some of the experimental data which I am trying to demonstrate and show you here. And we also have many references from different companies. And with this, I don't want to take you much of your time. Thank you very much. If you have any questions, so we can discuss later in the tea time. Then whatever the doubts you have, you can discuss with me. You can explain them. Thank you.
Thank you, Mr. Rajesh. And now we'll have a short 15 minutes break to attend the National Day ceremony outside.
FAO is defined pesticide as a substance or mixture of substance intended for preventing, destroying, or controlling any pest, including vectors of human or animal disease, and unwanted species, plants, or animals causing harm during or otherwise interfering with protection, processing, storage, transport, or marketing of food. Also, agricultural commodities, wood and wood products, or animals feature stuffs or substances that may be administered to animals for the control of insects, archaeans, or other pests or other bodies.
Sorry for that. Yes, no, it's okay. Sorry for that.
Okay. Pesticide residue in food can cause significant health risk. In this presentation, we will explore the impact of pesticide residue in human health and disease, and ways to reduce this exposure. First, pest. Pest is any harmful, destructive, or troublesome animals, plant, or microorganism. Pesticides is of any substance or mixtures of substance intended or prevent, destroying, repelling, or mitigation for any pests. Are different pests that destroy crops in agriculture, and to make control for these crops, we should to produce or use different type of pesticides or different chemical compounds to make the control in the crops or in any types of food may it be contaminated with pesticide. If we talk about weeds or any plants, we will find hundreds of this type under weed or unwanted plants. That means we should use many different types of pesticide, quantity, and huge amount of quantities and types of this component, chemicals component. Also, if we talk about insects, thousands of insects, that means also thousands of, maybe hundreds of thousands of pesticides used to make control, rodent, and also the fungi. Why do we need to use pesticide? In all agricultural countries, there are used pesticide to make control of the crops. They use it under control and law used in all these countries, mainly for 3 reasons. One of them is to protect crop loss, need reduction, protection during storage, and also vector disease control. Types of pesticide. As we say, there is thousands of types of pesticide, and this type of pesticide is, if we can imagine how many types of pesticide we are using in our crops, on the final products also during the storage or transportation, we will find we have huge number of this type. And according to that, this pesticide classified in 3 types. Herbicides, which is herbicides control weeds and unwanted plants in agriculture to prevent them from competing with crops for resource. Also, there is another group, insecticides, where the insecticide is target and eliminate harmful insects that can damage crops, reducing yield losses and ensuring plant health. The third type is fungicides. Fungicides also controlling fungal infection in crops. They help prevent diseases and improve overall crops quality and marketability. I don't want to scare you. The pesticide is available, but under the control. We analyzed many samples and we didn't find some of them is free from pesticide. Some of them is within the MRL, that means within the standards, and there is no effect. We cannot say no effect. There is some effect, but we can by some factors we may control the affecting the presence of pesticide residue in food. We can do it if we use agricultural practice, good practice in the farm. Agricultural practice. the types and amount of pesticide use as well as frequency of the applications and influence the presence of residual pesticide. Storage and transportation also is one of the important factors. Sometimes the pesticide is accumulated during the storage and transportation, particularly if there is no proper handling, temperature control, measurement are not implemented, maybe we will find also pesticide. Food processing method. Some of the food processing method may not effectively remove or degrade the pesticide residue, leading to that resistant in the final products. Resistant of pesticide in the final product. Potential health. There is many scientific studies of the potential health effects of hazards, chemicals, and pesticide. All of them to be classified as carcinogenic, can cause cancer, neurotoxic, can cause damage to the brain, or teratogenic, can cause damage to the organs. This process of classification is called hazard identification, and this is the first step of risk assessment. An example of hazard identification in classification of substance according to the carcinogenic to human, carried out from the International Agency of Research Cancer, AIRC, the specialized cancer agency of WHO. Same chemicals can different effect, different doses, which depend on how much of these chemicals a person is exposed to. It can also depend on the routine by which this exposure occurs, ingestion or inhalation or by injection. Health risk associated with pesticide exposure. Some of these pesticides have effects, short-term effectiveness, immediate health effects of pesticide exposure, such as skin irritation and respiratory issues. Another category have long-term impact, potential long-term health. This risk including chronic disease and reproductive disorders. The weak population, like children, pregnant women, and elderly are especially susceptible to the health effect of pesticide exposure. What are the dangers of this pesticide? Some possible reactions are gas, skin irrigation, noises, vomiting, breathing problems, brain disorders, plus disorders, liver maybe also is affect liver, kidney damage, reproductive manage, cancer, and sometimes it leads to death. Because there is a huge number of pesticides and the effect of these pesticides is different than the other. Globally, they are labeled this pesticide in 4 categories. The red one is the extremely toxic, like zinc phosphate and ethyl acetate. And this classified as a lethal dose. For the extremely toxic, the lethal dose from 1 to 50. The second label in yellow, highly toxic, from 51 to 500. The third category is the blue one, moderated toxins, from 501 to 5,000 lethal dose. And the slightly toxic, classified and labeled in green color. This is more than 5,000 lethal dose. Regulation and monitoring of the pesticide residue in food. Government agency establish regulation and monitoring the monitoring program to ensure the pesticide residue in food are within safe limits. Also, we have control by maximum reducing limits, MRL. These limits are legal limits set for specific pesticide in different food products to ensuring their safety for consumption. If we talk about monitoring, this is the main role of the laboratories testing. The laboratories analyze the samples of food and other types of products to detect and quantify the pesticide residue to ensuring compliance with regulation and within the MRLs. Workflow of the analysis in pesticide residue. This is very long procedure we handle in the lab, and we can categorize in 3 stages. The first stage is the sampling during the inspection, and this stage is, yes, this is the first stage, but it's very important stage because according to the sampling, we can control and identify from where maybe the pesticide came. Also, before analyze the sample, we should have representative sample. Second stage is the sample preparation. We use culture method in the lab. The third stage, the samples is analyzed with different instruments, very high accurate instrument, GC-MS-MS and LC-MS-MS, and we have different types of brands and different types of instrument, and all of them we are participate in BT samples, and we have accurate result. We analyze the samples. We have 2 labs, one location in Abu Dhabi and another one in Al Ain lab. The third and important also step is the result evaluation of the result and reporting, and this according to UAE regulation, codex, and EU regulations. Okay. Reducing exposure of pesticide residue, there is many tips to minimize and to reduce pesticide residue in the food, like eating variety of fruits and vegetables to minimize the potential of increasing exposure of single pesticide. Also, wash all the products, even that which is labeled organic. Nothing to wash, but we should do wash it even if it's organic. And this and that which you plan to clean. Also, we should wash our products under running waters rather than soaking or dunking it. Drops with clean clothes, towels or a paper towel when possible. Also, the scrub firm. Fruits and vegetables like melon and root vegetables. Discard the outlier of the layer of the leaves vegetables, such as lettuce and cabbage. Peel fruits and vegetables when possible. Also, drain fats and skin from meats, poultry, and fish to minimize the pesticide residue that may accumulate in the fats. Abu Dhabi Quality and Conformity Council have big capabilities in this field of pesticide residue in food and environment. We have 2 locations to analyze pesticide. One location in Abu Dhabi lab, in Mastar, and the other one in Al lab. All our work is accredited procedures under ISO 17025. In pesticide fields, we analyze 15 commodities, and the accuracy of our result and detection limit is below 10 ppb. Actually, we reach to 5 ppb, but we're reporting according to standard below 10 ppb. For the instrument, we have 5 GC-MS-MS, 4 LC-MS-MS, and we are the only lab in UAE who can analyze more than 702 pesticides in 15 commodities: fruit, vegetable, taste, honey, dried fruit, animal feeds, the tablets of animals feed and also the grasses, all types of cereals, tea, species, all types of milk and milk product. We are the only lab or the first lab analyze pesticide in egg, meat, all types of fish and seafood, and oil and fats, water. And some of this community, like the fish, oil and fats and egg, is very complicated matrix, but we are capable to analyze with below the 10 ppb, and we have participated in different programs to check our accuracy, and all we are passed. Pesticide test in ADQCC. If we took a look from 2021 until November 2023, we find increase the number of the samples that we are tested. This more than 50% from 21 to 2023 November. The increase of the number of pesticides is due to our government guides to provide safe food in UAE to our who live in Abu Dhabi and who are UAE. All the laboratories in all UAE pair together and cooperate to ensure the safe food for our residents and visitors. Thank you.
Yes. Yeah, I was mentioning that this, for me, one of the most important slides, because we should see the new technologies as a new tool, a really helpful tool. We should see as a partner, not as a robot that is going to replace our work. So as we can see on the picture, we are shaking hands, the robotic arm and the human arm. So okay, let's start with the presentation. First of all, what is lab automation? The first question that came into my mind is what are the real options to reduce the error and maximize the efficiency in our labs? So we can describe laboratory automation as a strategy to research, to develop and optimize technology in a laboratory and improve or update old methods. So using automatic technologies such as robotics, computers, or softwares, we can minimize the human errors. Many labs use automation to replace manual tasks, very repetitive tasks, and increasing the throughput and improving the quality. The direct benefit are the increase in the reproducibility, more accuracy in data, better efficiency, and also the traceability. So here I summarize the challenges on the lab. For sure, there are more challenges that you are facing every day on your labs. But the first one is one of the main concerns in the lab is the repeatability. We need to ensure reproducibility and accuracy in our lab results. Another one is standardized protocols. Sometimes we need to share different methods and results between different labs. So that's really important to have this tool to share this information between different labs or even different companies. Also, traceability and compliance is an aspect that became more important every day because we are working in a high regulated environment. So with water solution, we ensure that we are working under these traceable measures. Another one is our daily basics, the reproducibility and efficiency. For example, we are having everyday new projects. We have more samples in our labs, Many times we are the same number of users in the lab, so we have more work pending. So with these automation systems, we can reduce the cost per sample, and also we are going to reduce the time. So it's also make to have our resources in a more important task. So our users can work in a more useful task, such as researching, and the robot will be pipetting, doing the sample preparation. And last but not least, also important ergonomics. I saw many cases of injuries due to repetitive work doing the pipetting. So we can avoid this type of injuries with the automation. So I want to show you what are water solution that includes 3 pillars. The full solution name is Andrew Alliance Technology. There are 3 different parts, 3 pillars. The first one is One Lab. One Lab is the brain, is the software that control and enable total lab connectivity. It's really easy to use. I will show you how easy we can create a new protocol with this software at the end of the presentation. And also, as I mentioned before, is compliant with the regulation. The second part is pipette, that are the heart of the solution. There are controlled smart pipettes connected by Bluetooth that can improve the productivity and reproducibility. And the last part is the arm, the muscle of the solution. Andrew is the big muscle and the motion on all the process. So Andrew was designed to allow us to forget about the technology and focus on science. So let's move to the body. Andrew Plus is a cloud connectivity device. So the software is on the cloud, and we can connect the system to the cloud, and we can control our method from our laptop or our tablets on the lab. So providing reproducibility in fully traceable experiment for complete our workflows. Andrew can be installed really easy. Anyone here is able to install it. We have a remote team that is going to give you a remote support. Also, we have here a big team that is going to give you all the support. We have this demo unit here based in Dubai Lab. So the robot itself has 2 arms. The left arm is able to move around all the lab bench. You will see later. And the right arm is fixed. So it's going to hold all the pipette and the different grippers and devices. This Andrew robot is compatible with single and multi-channel pipettes from 0.2 microliters to 10 milliliters. So those are the pipettes, are controlled through the software One Lab, and there are the smart and electronic guided pipettes that are the manufacturer is Sartorius. These pipettes are charged in less than 1 hour, and they it's really easy to connect to the software. Okay. There are also some dominoes. In these dominoes, we can put our plates, microplates, waste plates, different. Well, you will see later on the software the different tools that we can add to our workflow. There are also some reservoirs for the solvent, different column and reaction blocks, and different kind of tubes. For microtubes, 15 ml tubes, 50 ml, and also for bottles. So we can adapt in total 11 different dominoes, And the footprint of the software you can see on the table right there. It can be installed in laminar flow hood and also in the biosafety cabinet. But your workflow might require more than just pipetting. So we can also include some electronic devices like the shaker glass that can allow us to shake our samples. We have it for microplate and also for the tubes. Another device is the Peltier that can allow us to cool the sample to 0 degrees and to heat to 99 degrees. It's available for microplates and also for 96 well plate for PCR. We have also the option of a magnet for 50 ml tubes and also for the PCR plates. And the last one is the extraction device. We have it there on the back behind the robot. I don't know if all of you can see the extraction device over there. With this device, we can automatize the sample preparation, all the solid phase extraction. So this is the extraction glass device. Is integrated on the full solution of Andrew Alliance. And with this device, we can process any sample with 0 hands-on time. It is connected to a pump, and there is also a bottle to collect all the waste. This device is compatible with macro and micro-elution plates, and also with cartridges from 1 ml, 3 and 6 ml. We can control the pressure profile. I will show you later. And there is also one gripper to move all the plates. When we are working with 1 ml and 3 ml, we can have 24 samples at the same time. And with the 6 ml cartridges, we can work with 12 different samples at the same time. About OneLab, the software, the head of the solution, is a unique software that can control all the automation procedure. We can run it on a PC or a tablet, and it's a free of charge software. So I invite you, all of you, to go into onelab.andrewlyons.com. You can register and you can see all the solution. You can create a protocol on your devices, and it will help you to guide your sample preparation. So you can define and execute many experiments. There is also a library with many applications that we already developed. I will give you some examples right now. So it's really easy to use, very intuitive, and you will see a graphic protocol. There is an online bench on the application, and we need to match with our bench on the laboratory. So what are the main applications of Andrew Alliance? The first one is the easiest, is I read for pharmaceutical industries that are repetitive and manual pipetting. We're starting with serial dilution, concentration and normalization. We can work with the viscous samples. Also, to avoid the contact with hazardous solvents, we can install it in a cabinet. Also for standard course preparation and for plate reformatting. All these different tasks can be done also in food and environmental applications. The advantage of automation in food and environmental lab is that we can include all the solid phase extraction. We can also have the clinical workflow for DNA preparation, ELISAs, and also for some kits. In biopharma workflow, we can have kits for amino acid, for glycan, for protein digestion, protein precipitation, and peptide mapping, for example. And in conclusion, human error are always there. So with the automation, we can have many advantages, such as increasing the reproducibility and efficiency on the lab, reducing the time and also the cost. It's fully traceable. The software will record every step that we've done on the lab. We can create different lab users and it will be tracked. It will also reduce the training needs. So sometimes we don't have time for trainings, or there are many people working in the lab. There are variations on the personnel. It's very boring task. We can have a random mistake. So with the automation, we will avoid all of these problems. And for me, that's important. We leave more time for the important task. So now I will show you one example. What we are going to do now is create solid phase extraction with 3 ml Oasis HLB prime cartridges. And it's not necessary to equilibrate and conditioning the cartridges. So we will load the sample, then we will wash with the solvent, and then we will elute and collect into the vials. Okay. This is the...
You're offline, so that seems that you will not be able to do the demonstration now because otherwise we have no Wi-Fi.
No.
It works?
Yeah.
Okay, so let's try.
Let's disconnect the phone. Zoom. But we are connected to Wi-Fi 1. Connected to both at the same time. Yeah, that have Zoom things. Okay. Okay. Do you want to do? I want to show you. Share your screen. Okay, which one is it? This one? Okay.
Okay, let's continue. So what I want now to do is create a new protocol. You will see how easy. I'm sure that all of you can create a solid phase extraction protocol. So this is how our lab bench looks like, our online bench. So here, this icon, we can click, and it's going to show us all the labware available. From microtubes, Falcon tubes, plates, different vials, different volumes. So now what we are going to do is to create a protocol. We will have our samples. We have 2 samples in these microtubes. So we add 2 microtubes to the lab, to the bench, another Falcon tubes for our solvent. And now I'm going to filter. I can click here for 24 samples. I will use Oasis HLB, in that case will be prime. I add it to the bench, and also vials for collecting the samples. So now we have all our lab. We're here on the right. We just click on add to bench. And this is our online bench. Yeah, clicking on these arrows, we can move all the labware through the lab bench. I really love to organize like this. Okay. So now what we are going to do is to label the different labware. We are going to add sample. We name sample 1. We click here, again, fill with a reagent. Sample 2. Then we are going to click on the solvent. 5% methanol in water for the wash, and for dilution, we will add 90% acetonitrile and 10% methanol. So now we click on the pipette and we just select the different wells. In that case, we are going to duplicate the sample. We are going to add to the well D1 and D2, save selection. We are going to add 0.5 milliliter. Those are the advanced pipette options. We are going to do in forward pipette mode. We can also select the aspiration and dispensing speed. We can create some bubbles inside. We are working with viscous solvent, and also the tip position. In that case, we will take it with respect to the liquid, and for the density, we will drop on the fly. It's going to be the same sample, so we are not going to change the tip between the different steps. We are going to save tips. Save a step. Now we're going to do the same with sample 2. In that case, to well D3 and D4, save selection. And again, we fill with 500 microliters. We will dispense on the fly, and we're not going to change the tip between the pipetting steps. Now we save the step. And what we are going to do is to wash our samples. We are going to select the different wells, in that case from D1 to D4. Save the selection. We are going to add 1 milliliter of the wash elution solvent. We are going to dispense on the fly, and we are not going to change the tips between steps. Now we are going to do the vacuum. We are going to extract. We just click here on actions. I will do it again. It was so fast. We click here in actions and flow through extraction. Just clicking here, we are going to drop the plate, this domino with the Oasis HLB 3 ml cartridges. And in that case, we are not going to collect the samples, so it will go direct to the waste. So here, that's really important for me, is that we can create a pressure profile. We are going to start with 2 millibars for 10 seconds. We will add a second step of 3 millibars for another 10 seconds, and the last one, 5 millibars for 5 seconds. You can see here the pressure profile that we have created, but we can add many steps. It's going to be a quick method, so I don't want to spend more time on the extraction. We create the pressure profile, we save the step. So what we are going to do now is to elute our analyte. We select the elution solvent. We will drop it on the D1 to D4, save selection. Again, we will add 1 milliliter, the destiny on the fly, and we save the step. Now we are going to do exactly the same. We will click on action, flow through extraction, and now we will drop the extraction labware, our cartridges, and in that case we are going to collect, so we will drop the vials. So now what we can do is to extract without any profile for 30 seconds at 2 millibars, and we save the step. So now we complete the workflow for the solid phase extraction. So what we are going to do now is clicking here on execute. And the software is going to calculate how long it will take to do all the process. We have it manually. We are going to spend around 9 minutes if we do the full workflow manually, and Andrew will last less than 7 minutes. So we click here, and it's going to show us all the material. Andrew, the robot, the extraction device, is going to use 1 milliliter pipettes, and also this gripper, the tips that the robot is going to use, and the different blocks that are already placed on the bench. And here, the labware. So we click and continue. But this is the view of all the different dominoes that we are going to use during this experiment. Those are the tips. Second one are the vials for the collection. Here we have the microtubes with the sample. We need to confirm that we have the right volumes on the right position. And here we have the solvent that are also correct. And here is the extraction plus device. So we need to be sure that everything is in the correct place. And once we check it, we start experiment. So now Andrew will start to move. So you see there is another eye over there. So what it's doing now is checking all the barcodes that the Domino's has on the top part. It's going to double check if everything is on the right position. Now it's checking the 50 ml tubes. If everything is correct, if there is something wrong, the process will stop and I will have some notification on the screen. Now the check-in is complete and it's going to start with the workflow. Now it's taking the 1 ml pipette. So what it's doing now is checking if the pipette is calibrated. It's going to do it the first time they use a pipette. And once they take the tip, it's going to do the same process. It's going to see if everything is correct with the tip and the pipette. Then when the robot do the check-in, we'll start with the pipetting process. Taking the tip, that's normal. And now it's taking the tip. It's going to double check if everything is correct. Over there is a laser and a light. You cannot see from your side. So now everything is correct. It's going to take the samples and place it on the tubes. Take another 6 minutes. So I will ask you if you have any question, suggestion. exchanging the tip because we are going to have another sample. Yeah, on this side, you can see all the different steps, the time remaining. Question? No.
Okay. I'll switch the. Share. Thank you, Mr. Alvaro. Please join me in welcoming Mr. Abdullah Siddiqui, Head of Research, Technical Consulting and Training Section in Abu Dhabi Quality and Conformity Council, who will speak about the leachable and extractables in food contact materials.
Assalamu alaikum, everyone. My name is Abdullah Siddiqui. I am Head of Research and Development Lab, comes under the Technical Affairs Department of Central Testing Lab, Abu Dhabi Quality Conformity Council. The topic is leachable and extractables in food contact materials as well as the pharmaceutical. And the CTL capabilities to investigate different cases. Also. Okay. So this is a common challenge for industry, regulators, and even scientists, because the leachable and extractable is a big issue for any kind of products. Either this is pharmaceutical, medical devices, food, cosmetics. All the quality of the product is at the end depends on what type of packaging material industries are they using. So leachable and extractable, these are two different words. Leachables are chemicals that migrate into food or in drug from the packaging material and in a closed system. It could be polymer, lubricant, color pigments, dyes, heavy metals, organic volatile impurities. It depends. Then extractable Okay. Here. Here. Okay. Here we go. So it's a growing challenge for all industries. Now, if we want to understand the extractable and leachable, when we talk about extractable, this means we are talking about the materials. This is a common example of inhaler. When we want to extract from the body of medical device, this will come under the extractables. Where the leachable, when we talk about leachable, we are talking about the product. Why investigations are important? It's a growing concern of industries, as I said. It's important to control product quality, product stability, product shelf life. Assigning of product shelf life even sometimes depends on leachable and extractables concentration. And if you want to improve the quality of product, we have to work on packaging material. So again, packaging material is directly related to leachable and extractables. To calculate the health risk factor, also such kind of studies are very important. Unfortunately, no single regulatory methodology is available from any top regulator in the world, FDA, WHO. If you want to plan any packaging related study, leachable related study, you have to take so many methodologies together, and then you have to design your own protocol to conduct such studies. PQRI is one organization. They are trying to compile all these different regulations, and this is internationally acceptable by the different industries. Major group of leachable and extractables. There are many things when we talk about the leachable and extractables. There are 8 or 9 different major groups, which comes under the volatile organic compound, semi-volatile, non-volatile, elemental impurities, identification and quantification of contaminants, known and unknown, PFAS, which is very hot topic these days, particulate matters, and overall migration. So these are different topics we have to cover whenever we are planning any packaging related or food packaging or pharmaceutical packaging related study. I'm from CTL. The capabilities of CTL investigation such cases. We have state-of-the-art analytical facility in Abu Dhabi and in Al-Ain to conduct and to support such scientific studies. We have in-house setup for chemical and microbiological analysis. We have radiochemistry setup, accredited radiochemistry setup. We have characterization and leachable, extractable capabilities to investigate, chromatographic and spectrophotometric investigation, stability testing, product intercomparison studies, and we have also capability to calculate the health impact at the end. These are the challenges faced by different modern industries these days. Selection of the right packaging material. This is one of the challenge. Identification and quantification of phthalates. This is very important. Again, comes under the PFAS. Overall migration load, presence of additives and their identification, particulate matters and their sources, polymers and its characterization. This is again another one of the most hottest topic of the current world. Bisphenol A and stability of packaging material. These are the challenges normally the modern industries are facing these days. To support industry, CTL is equipped with, we have all kind of instrumentation available in central testing lab. We have lots of HPLCs with UPLCs set up, GCMS with pyrolyzers, LC-MSMS, GC-MS, FID, ECD, TCD, and other detectors, GC with double mass, GC with head space, purge and trap, ICP-MS, ICP-OES. EDX, RF, flow cytometry. These are all the equipments are available in central testing lab, and very high utilization on different projects. FTIR, IR microscopy is required for the identification of any unknown molecules. These days, one recent achievement of CTL related to leachable and extractable. Recently, we have concluded one very interesting study. This study is for paper cups, the coffee and tea paper cups. It was very surprising for us that why it's a paper cup and there is no seepage and no leakage, because this is unrealistic that I have paper, I transfer or I pour a huge amount of hot liquid, but there is no seepage and there is no leakage. What is the reason behind it? So this project came to us recently. We have concluded. I want to share with you all the findings and the experimental work, what we have done during this study. A detailed study have been conducted recently on commercially available paper cups in Abu Dhabi market. Local and international standards we applied to investigate the quality and the safety parameters, including UAE GSO 1863, GSO 2231, European Union 10/2011, and ENBS 11869. So these are the most important regulatory parameters we have applied on paper cups. Main objective of the study was to assess the safety concern about the paper cups, focusing potential human and environmental impact. This study helps to understand the reason of water retention in paper cup. Aim of study was to identify and quantify the leachable and extractables from the paper cup, that how it's going to harm human health. To conduct this test, we have conducted many group of investigations, including overall migration. We also work on specific migration. Under the specific migration, we have done so many tests, like, for example, we have done the toxic organic volatiles, phthalates, plasticizer, microplastic migrated from the body of cup to the liquid, identification of hazardous material in paper cup, test for the presence of any polymer, and characterization of any unknown material which we found during the test. Method and instrumentation applied. We applied all state-of-the-art instrumentation during this study to perform the investigation. GC-MSMS, flow cytometry, Fourier transform, infrared mass spectroscopy, GC-MS with Persian trap, IR microscopy. Study extended to target health hazard and to prove leachable and extractable chemicals migrated from suspected hydrophobic layer of paper cups to the drink. Because the surprising area is why this is hydrophobic. If the claim is this is a paper cup. Test for overall migration, we followed UAE GSO 1863 2021 and GSO 2231 standard. And as we followed exactly the protocol, 70 degrees centigrade, we prepared the tea and we place, we pour in cup and we kept it 70 degrees centigrade for 2 hours. And after 2 hours, we dried the residue and we tried to see what is inside. So all type of different brands we collected from the Abu Dhabi market, and we found almost 100% different type of samples fit for use. The maximum residual acceptable limit is 60 milligram per kg, and we found very minute concentration. This will come here. So the maximum concentration we found it's only 4 milligram per kg. So this was amazing, because what we have seen that this is paper cup. When we transfer or pour the hot water, tea and coffee, we found nothing inside. This means nothing is coming out from the paper body. So all considered pass. This is for overall migration. Then we went for a specific migration. In a specific migration, we prepare, because the article is intended to use for coffee, cup, gawa. We prepare fresh all these products in our lab, and we pour in these cups, and we kept as per the protocol, and we found nothing. the 30 different health hazard parameters we targeted by using GC MSMS and GC Headspace. Means it was again surprising for us. So what is inside? Means nothing is inside. No overall migration. This is surprising. No specific migration. So what is this cup is? This the results. We got it. Then we have done some forensic study on paper cups beyond standard. What we have done, we kept these cups in water, millipore water, organic free for overnight. And then next day we peeled out all the water, all the layer of paper, slowly with hand. And then we found a clean, thin plastic layer embedded inside the paper cup. So this means that this is not a paper cup. It has a proper lining, thin, shiny plastic material inside. Then we went for further. We tried to check the diameter and the thickness. We found that the total paper cup, on average, it was 0.5 mm. And out of 20.5 mm, 0.04 mm was the plastic inside. So which is 8% of the total thickness of the paper cup. We checked what is this type of plastic, and we went for FTIR IR microscopy and confirmed that this is polyacetylene, which is an organic polymer. So all brands collected from Abu Dhabi market, different type of paper cups, all brands found positive with polyacetylene. Then we went for extractable analysis. From here till now, it was all leachable. Then what we have done, we took this plastic and we went for further analysis of these plastic, what we found inside the paper cup. And we went for phthalate analysis. We found nothing. This means that the plastic itself is a safe plastic. So we targeted 7 top suspected phthalate, which is plasticizer, DIBP, DBP. Out of 7, 4 we found positive, but very low concentration, where the acceptable concentration is around 1000 ppm. So it's around 0.1% of, we found only 0.01, 0.02, 0.02, that's it. So it was negligible, you can say. Then we tried to check, is there any disintegrated part of this plastic which is embedded inside the body in water, like microplastic particles through FTIR and flow cytometry. We found nothing. This means no even particles is coming out from this plastic. So conclusion is no evidence of any targeted leachable from tested paper cups. Overall migration test found within the limit. So this means that it's pass for overall migration. No evidence of phthalate found. Maximum we found 1 ppm. No evidence of plastic microplastic particles even. No evidence of human hazard chemicals. Hydrophobic nature of paper cup is due to polyacetylene. This is a direct threat for environment because this is non-recyclable. This is a big challenge, means it is not going to harm human health, but seriously going to harm environmental health. This is very another topic for you. So, yes, so it was surprising for us because this is organic polymer. We don't have data of UAE, but we have data of UK. It's around 2.5 billion paper cups they are using per year. This means that 7 million paper cups per day and 5,000 paper cups they thrown in dump as per minute. This means that whatever we are throwing in recycling bin, it's not a recyclable. It's not a recyclable because this is embedded with, it's so difficult to separate plastic from the paper. Then as our suggestion is, we have to educate industries to use recyclable material, and we have to go for health impact on nano level, because we found nothing on micro level. We have done up to ppm and ppb level, but not on femtograms or picograms. So this is our conclusion. Thank you.
Thank you, Mr. Abdallah Siddiqui. And now let's have a short 30 minutes break for prayer and coffee. Thank you.